All right, we're taking care of two processes in one go, transcription and translation. They are a part of the central dogma of biology, which explains how it is that your DNA can be changed into mRNA, and then that mRNA can be translated into a protein that ultimately gives you all of your traits. So, starting things off, there are a few things to look at here. The original DNA strand, which is sort of running along the bottom, is a gene that has a promoter at its beginning that lets things know where to start, a region that is coding the gene, and then a terminator, which basically says that it's finished. RNA polymerase is floating around with random molecular motion. It attaches to the promoter region and then runs down the gene. Now, as it does this, it is attaching these free RNA nucleotides that we can sort of see out here, just kind of float around. It takes them and attaches them based on the lower strand here and creates a complementary strand of mRNA, which we can see sort of snaking out here. The further along it goes, the longer the mRNA strand will be. Again, it has to attach things to the 3' end of a strand, so just like always, DNA runs from 5 towards 3'. It's worth noting here that the RNA polymerase has to look at two strands of DNA. There's a coding or a sense strand, and then there is a non-coding antisense or template strand. So taking a look at the two DNA strands down here, I've just made up a couple of codes. Ultimately, what you want RNA polymerase to do, you can sort of see it behind the scenes here, is you want RNA polymerase to build complementary to the antisense strand. That way, the code represented up here in the mRNA will be identical to the sense strand. Again, if you build complementary to the antisense, you get a strand of mRNA that is identical to this code, which is the sense strand, it's the coding strand of DNA. That's the code you want in the mRNA. So, you may remember that you need to remove some introns from that mRNA so it can become mature, splicing the exons together with a little spliceosome or a SNRP. And you might remember that tRNA is coming up here. tRNA has an anticodon region. It's got these sections where the RNA has base paired to itself and loops where it has not, 3 loops to be exact. And then there's an acceptor loop up here at the top that actually has an amino acid attached to it. You can have more than one ribosome running along this mRNA, which is the next process, translation. If you have more than one on the mRNA, it's called a polysome. And so here we are. This is translation. Now, I know there's a lot to take in here, which is why there's a little GIF that's going to show the process. The ribosome assembles around the mRNA. And then these little blue objects that you can sort of see right here are the tRNA. The tRNA's anticodon binds with a codon on the mRNA. And based on that codon, it is going to leave behind the appropriate amino acid. So looking back here, you can see on the inside here, this anticodon will match to a codon here, A to T, G to C. Or I'm sorry, there's no T, it's only U now that we're talking about mRNA. A, U, G, and C. Based on that, it will make its way through the ribosome, leaving behind an amino acid that will be connected to other amino acids, slowly forming a polypeptide that is going to snake out the top. It's also worth remembering that there are a couple steps here for the ribosome: initiation, elongation, translocation, and termination of the polypeptide. And that ultimately what is happening up here is you are creating the peptide bonds that you can diagram with individual amino acids. The creation of this peptide bond is exactly what the ribosome is catalyzing. And in addition, ultimately, if these ribosomes are attached to the rough endoplasmic reticulum, it's worth noting that they are going to make their way to the Golgi apparatus. And then from the Golgi apparatus, they will leave the cell through exocytosis, because that's just what cells do with proteins that are made in the rough endoplasmic reticulum.